Welcome. It's my pleasure to welcome Rohith Krishna, who's going to lead us in a discussion on the RoseTTAFold All-Atom model. Rohith, maybe I'll let you introduce yourself and where you're coming from, and we'll hopefully get into a good discussion.
Yeah, thanks. Thanks for having me. My name is Rohith. I'm a graduate student in David Baker's group at the University of Washington. I'm super excited to share some of the work that we've been working on for a while now. Um, Obligatory before getting into the details is that this is not just my work. This is work of many, many people, and this would not be possible without all of these people on the slide. So that's just something to keep in mind throughout the talk, that many great ideas came from many different places, and it's really exciting to be able to collaborate with so many people and learn from so many people. So the general idea of what I'm going to talk about splits into two different tasks, right? The first being structure prediction and the second being backbone generation. And this is probably review for most people in this group, but for structure prediction, you start with an amino acid sequence, and then you want to produce a 3D structure. And then backbone generation, in protein design, you often start with a corrupted structure. So either a fully corrupted structure, we have no idea what is in it, or a partially corrupted structure where some motif information or some privileged information about what type of structure you want. And your task then is to recreate a 3D structure. And so, as you all know, deep learning has had a huge impact on this concept of protein structure. And this really, and I'm not going to go through the details here, but this really hinges on two main things that were introduced in AlphaFold 2, right? The first is this idea of inductive biases that are problem domain specific. So, for example, in AlphaFold 2, you have these pairwise attentions that get biased by a third node to enable attending over triplets to enforce the triangle inequality of distances. And there's also the second idea of data set augmentation. So, the data sets we have in biology and structural biology are smaller than those for, say, images or text. So how do we kind of be clever and augment these training data sets? So the other thing is that when we think about protein structure prediction, it's really an underspecified problem. In the PDB, there's much more richness in terms of protein multimers, protein nucleic acid complexes, protein small molecule complexes. And systems like AlphaFold 2 and RoseTTAFold are actually really resilient to the lack of full context. So this is an example from the AlphaFold paper where they show this really accurate prediction despite not knowing the other two chains in the assembly. And so this is really exciting, right? Because you can make all these accurate protein structure predictions kind of without knowing this auxiliary context. But what you do lose in the process is you don't know how those auxiliary molecules are interacting with your predicted protein structure. And so many people have tried to remedy this by taking things from the AlphaFold database and then adding on things such as post-translational modifications or ligands post facto. But this is really was, at least when I started working on this, was kind of a problem that felt like it was not being adequately resolved. But now there's been tons of work by many people to this end. And so the kind of impetus of this work was, can we design a neural network architecture that actually explicitly represents all that functional richness that's in the PDB? So you think in the PDB, you have obviously proteins that we've gotten really, really good at predicting, nucleic acids, metals, small molecules, and covalently modified proteins. So we really want to think about how do we develop this architecture with all of these molecules in mind, and likewise the loss functions and the training data sets. And is there a way that we can kind of create a unified network architecture that can model everything? And the other big motivator for us is that protein design really requires the specification of the ligand. So this is work by my co-author Woody Ahern in his previous paper, where he shows that if you're trying to create a small molecule binding protein without knowing the small molecule with RFDiffusion, you really run into a lot of pathological problems, right? So one, perhaps obvious problem is that you get proteins that don't really have a pocket. So they don't really have good shape complementarity to the molecule. And the second, which is even more problematic, is that the backbone just clashes with where your molecule would go, right? Or where you intended your molecule to go. So you really need to have some sort of understanding of what is the small molecule or ligand that you're trying to bind to create these kind of ligand binding proteins with the latest tools that we have. And many applications of protein design require ligands. So not to get into details here, but for things like enzyme design, for small molecule diagnostics, or on the right is a really cool application of creating this like photosystem special pair, right, where these two chlorophylls are coordinated in a very specific geometry. So this is really something that we felt like the deep learning methods hadn't really impacted the way they had for the protein only case. And so the other intuition that we had in the lab was that these structure prediction models and the backbone generation models share many similar characteristics, or a good structure prediction model and a good backbone generation model share many good characteristics. And so one example is this idea of like sc3 equivariance, where if you give an input structure, the update should rotate, and you rotate it, the updated structure should be with the same rotation, right? And the other nice thing about these models, or they explicitly model coordinates, and that's kind of like the problem outlined here. It's like you don't have an explicit model of the coordinates. So the model intrinsically doesn't have representation about what is happening with these extra atoms. And the other thing is both these models need this deep understanding of these interactions between residues or atoms in the training structures, right? So you could imagine learning things like polar interactions or things like shape complementarity, right? And the final thing is that they need these multiple inputs and outputs so you can do conditional tasks. So like I mentioned in the start, you might want to fully corrupt your protein and then create unconditional, just new scaffolds that didn't exist before. But in a lot of cases, when you want to do protein design with a specific function in mind, you actually have a motif or a small molecule per se that you want to bind. And so you want to provide that as conditional information. And so the kind of intuition going here, and obviously there are differences as well, but the kind of intuition is that if we can create a really good architecture for structure prediction, that this would have immediate application in design, especially with things like RFDiffusion, where the backbone generation model is fine-tuned from the structure prediction weights. And so there's multiple ways you can pose this problem. So now we're kind of getting into how you think about setting up the architecture, right? The first way you can think about this is this atomic representation, right? Where every atom in the protein and the small molecule and the DNA and all of your metal ions, et cetera, are represented as atoms in some sort of graph neural network. Or, I mean, it doesn't have to be a graph neural network. You could also be like an energy function where every atom has an interaction with every other atom. The second way you could think about it is kind of in this like sequential manner, right? First, you model the protein, and then you model in the small molecule or metal or whatever your desired ligand of choice is. And then the third is kind of this kind of idea of joint modeling, where you're both folding the protein and sampling the protein conformational ensemble and sampling where the ligand binds simultaneously. And so we felt, at least especially for the protein design case, that doing this joint sampling would give a very rich representation, which would allow you to sample new scaffolds that bind to small molecules. And so we went about creating this new architecture where you provide both an amino acid sequence and chemical structure. So just the 2D base chemical structure. So no privileged like structural conformation information. And then now you can expand your training data. So instead of just having proteins, protein residues, you can have protein residues or nucleic acid bases, as was shown in RoseTTAFold nucleic acid. And you can also have atoms. So you can have small molecules, post-translational modifications, all of these that are sent jointly into the model. And yeah, and so then now you open up if you could create an architecture to take these two inputs, you open up a lot of these different applications. And so now you can repose that same problem that I showed in the first slide, right? And so structure prediction, instead of just providing an amino acid sequence and getting back structure, you provide an amino acid sequence and a chemical structure. And then in backbone generation, you can corrupt a structure with a ligand, right? And then reconstruct the 3D structure with the ligand. And so those are kind of the two problem domains that we really set out to make progress on. So first, I'm going to talk a little bit about the structure prediction aspect. And this is where we mostly developed like the architecture of how the structure prediction, the neural network is encoded. And so we started with RoseTTAFold 2, and this is work from Minkyung, Baek, and Frank DiMaio. And so I usually kind of skip through the slide really quickly, but I think this audience might enjoy listening to some of the details. But basically, this model has 3 tracks. So there's 3 sets of representations that are updated iteratively over many, many blocks, right? The first is this 1D track, which takes multiple sequence alignment information and performs these like column-wise and row-wise attention operations on it to update it. And these are actually receive bias from the 3D track. So you create an embedding of the structure, you bias back the sequence embedding based on the structure embedding, and then you kind of do the same thing all around. So you take the 1D information, you bias the 2D information, which represents pairwise distances and angles. And then every step of this process, you're creating a structure. So every block, and in RoseTTAFold 2, I think there's 32. I think in RoseTTAFold All-Atom, there's a couple more. But in every block, you create a new structure. And then the predicted structure from the previous step is an input into the next step. So if you, so you iteratively refine your structure. And the architecture of the 3D step uses this thing called the SC3 transformer. So this is basically a neural network architecture that's equivariant with respect to rotations and translations. And I'll talk a little bit more in detail about that in a couple slides. So, okay, so now we're here and we want to go to creating a model that satisfies the restraints that I had outlined previously. And so the first thing you have to do, right, is that you need to add new tokens into the 1D track. So the 1D representation needs to functionally be able to distinguish what these two atoms, the difference between these two atoms. The second thing, right, is you need to provide some notion of bonded distance, right? When you have a protein, they're linearly encoded, right? So residue A, residue 1 is bonded to residue 2, and then you can provide some relative positional offset. But for small molecules, obviously that property isn't true. Like atom 1 could be bonded to atom 9, etc. So you need to provide some sort of notion of bonded geometry. And so the next thing, right, is you need losses that apply to small molecules. So in RoseTTAFold and AlphaFold, the losses take advantage of this NC alpha C frame that every residue has. But obviously, arbitrary molecules don't have canonical or the same frame, right, that you can use for frame alignment. And the other thing is that now the structure module needs to update the small molecule atom positions, right? And this also, at this point, we're still underspecified, right? Because you don't know the chirality of the molecule. So you need to provide the chirality as input features at some point in this network. And you can think the 1D and 2D representations are actually invariant to chirality. Like your distances are going to be the same in either chirality. So you need to provide this bias actually via the 3D features. And so the first thing you do, right, when you're creating a neural network is you need to have a training data set for it. So, and we basically went through the PDB and curated training data sets, and we clustered them by sequence. And so this is just kind of showing you the kind of details. This is also a table in the supplementary information somewhere in the preprint. And so, yeah, the one thing I'll point out is that, so for covalently modified proteins, there's a couple interesting things that come up, right? One is that you can have, in PDB entries, they're covalent bonds to proteins, but then they're also covalent bonds between small molecules, right? So you have to handle those cases as well. So there's a lot of data set processing and understanding the input data that goes into kind of making these training data sets work nicely. And the other thing we do, kind of building on this point that I had kind of pointed out earlier, is that we're often in this data limited regime, right? And specifically in the PDB, there's roughly 81,000 small molecules. And but if you cluster those with Tanimoto's score, with a very generous cutoff actually of 0.8, you get very few clusters. So we had this intuition that using the Cambridge structural database, which are crystal structures of small molecules, then we could augment the training data set of small molecules. And we really, and we don't really expect that this is going to give us like great improvement, but the idea is that you really regularize against overfitting on features that are only present in molecules in the PDB. And so,
before you go on, can I ask a question about this?
Yeah.
So you're not the first person to curate small molecules out of the PDB. So I'm curious on, did you consider using like PDB bind or biolip, which have already gone through and done this? And how would your curation differ from these other approaches?
Yeah, so we actually use PDB bind and biolips like filters in many cases. So, and we kind of make the lists. The reason that it's easier to, or maybe not easier is the wrong word, but the reason that we chose to do it this way is that it created a more streamlined data processing system for us. And you also, in PDB bind and biolip, oftentimes can lose some information about biological assemblies, like when there are multiple of those. And I think there might be encoded information about these kind of covalent bonds going across small molecules or cross small molecules to proteins, but it wasn't as clear how to extract those.
Will this curation be available when you release the model and weights?
It's hard. I think it's hard because the curation that we did actually isn't it's just taking CIF files from the PDB. So it's not really, there's nothing really there to release. I guess we can release the sequence clustering like with MSeqs, but I think
I guess maybe just the scripts that you processed it with. I guess why I'm asking is if somebody wants to do a head-to-head comparison, oftentimes you can't do that unless you're training on the same data sets. And if you don't know what the data set is, it's hard to compare.
Yeah, totally. Yeah, I need to consult with other people who are also working on these things. I'm generally of the opinion that things should be more open than not, but this one is actually it shouldn't be much. It's just downloading CIF files from the PDB. So yeah. So now kind of drilling a little bit deeper into the details, how does this actually kind of work out in practice? you have these sequence inputs, right? You have your sequences, you have your homologous sequences as per multiple sequence alignments, right? And then you might have homologous template structures for your protein. And you basically can combine these with element types, right? And then bond types, so you can give some sort of notion of bond order between atoms, right? And finally, this idea of chiral centers, and I'll get a little bit deeper into that as well. So this is just like a pseudo molecule that I drew, and this is the corresponding featurization that you'd get. So you see double bonds would give you some sort of different featurization than single bonds. Aromatic bonds also get a different type of featurization. And these are the atoms that can be represented. So they're fairly broad. Yeah, there might be one or two atoms in the PDB that we missed, but the definition is fairly broad. And yeah, so now the last thing is this idea of chiral centers, right? How do you encode chiral centers into the model, right? And so a little bit of intuition, right? So to create these 3D structures, we use a neural network architecture called the SE3 transformer. And this is built on a line of work called tensor field networks. And the general idea, and this visual is taken from this lecture, but the general idea of these tensor field networks is that they use these spherical harmonic functions to restrict the functional form of the filter of the neural network. And they have this interesting property that you can mix these like scalar and vector features based on this like L equals 0 or L equals 1 using tensor products and maintain equivariance. And so at each block of the network, we predict a structure update using this type of an architecture, architecture with this type of a functional form. And so we had this intuition that you can provide these like pseudo forces as vector features or L equals 1 features to the network. And the way you can provide these, compute these forces are developing these pseudo energy terms, right? And then just taking the derivative of the predicted of that energy term with respect to the predicted coordinates of the previous step. So you could imagine, right? You have a chiral center on the wrong side of a molecule, and the energy of that will be really bad, and the gradient will point that atom to the right direction. And so getting a little bit deeper just into exactly how it's featurized, if you kind of look here, sorry, there's a lot of things on the slide, but if you start here, O is your chiral center, A, B, C, D are your 4 different substituents. And then likewise, this is your other center where, you know, this is coming out and this is going in. If you compute this like dihedral angle, basically, where you use O, A, B, and C, you get a different sign depending on whether you, which one of these that you have, right? And then now you can take the predicted and the ideal. So then you can draw a unit sphere, circumscribe this tetrahedron inside it, and compute what the ideal angles would be. And, you know, in practice, obviously your chiral centers won't always be perfect, but this nudges them in the right direction. So finally, you'll kind of get your L1 features. In this case, we'll push A this way, and in this case, we'll push A that way. And yeah, we do this like very simply. We actually just use autograd, not the explicit derivative of the function, and it seems to work all right.
Sorry, can I ask one more question about this?
Yeah.
So intuitively, I think of the chirality as being discrete. It's either one or the other. But I guess this makes it, which is difficult to differentiate through discrete functions. But this, I guess, puts it into a continuous space in order to allow the chirality to be kind of continuous and therefore differentiable in order to allow for gradient updates on this. Is that sort of what the problem that this is trying to solve?
Yeah. Yeah, I think like when you think of chirality in a discrete manner, it also requires like some human intuition, right? of like how the substituents should be ordered. And we also, the intuition was kind of that we did not want the network to have to learn how humans have decided to define chirality. And this is a much more geometric description that we felt the network would like more.
Super cool.
The other conceptual thing that we want to introduce was this idea of atomization. And so there's a lot of previous literature that's discussed like the similarity of interactions between proteins internally and with proteins and small molecules. So we wanted to augment the data set by taking random chunks of proteins and then featurizing them as atoms, right? Because proteins functionally are comprised of atoms. And so in these cases, we kind of remove all this like multiple sequence alignment and template information and just provide the kind of aforementioned element types, bond, oh, sorry, you can't see my hands, element types, bond types, and chiral center information for these atoms, and then sample this. And this is, and I think this is a really a nice form of data augmentation, but really what this enables is prediction of covalent modifications, right? Because one potential issue with covalent modifications, if you have residues that are bonded to atoms, is you don't know which atom in the residue is bonded to which atom, or you might be able to say which atom in your like small molecule is generating a bond, but you don't know which atom in the residue is interacting. And this is because in these AlphaFold and RoseTTAFold type of models, only the backbone is explicitly represented and the side chains are represented as internal angles, right? So when you have this internal angle representation, it's very hard to distinguish like two atoms on a side chain. But what you can do with this atomized representation is you atomize the residue with the covalent modification, and then in the bond features, you can explicitly draw that covalent bond between the atom in the modified residue and the modification, the chemical modification. And you also obviously need to update the chirality information in these cases because you might be creating or destroying new chiral centers. And then you can, and so then the problem kind of arises that you still have the same problem, right? Because you need to know how where that residue is in the polypeptide sequence. And so we basically just did two things to do this. I didn't prepare a slide on this one, but just one is there's an additional bond feature that indicates backbone bonds. And then the second is the positional encoding gets both kind of residue and bond symmetry breaking information. So that now the network knows where in the polypeptide sequence that residue is.
So this idea of taking the residue level representation and treating it as an atom level representation, I think is super interesting. I'm curious if you already have 3 tracks, right, of having the harmonization among these different representations. I think what I'm understanding here is that you just used the atom coordinates from the training data as sort of, you treated it as this representation and then use that as part of the training objective. But I'm curious if you thought about it in terms of like this sort of self-consistency among these different representations as perhaps like a 4th track in this architecture, and if maybe you could do some sort of self-supervised or harmonization objective to make sure that these representations are giving consistent geometries during the whole prediction network.
Yeah, I'm not fully sure I understand, but so your point is basically like that you can ask the network to predict both the residue level representation and the atom level representation, and then have a 4th piece of information measuring their alignment or something like that. Am I understanding correctly?
Move back and forth between the residue and atom, like the frame versus the coordinates, as sort of moving back and forth between a sort of a 4th track in the architecture.
Yeah, that's an interesting idea. Yeah, we haven't explored that in particular. I guess, it's a super interesting idea. So sorry, I keep forgetting what the next slide is. So this is kind of the general idea of how the architect. I mean, there's some more details, obviously, but these are the kind of broad ideas of different how the architecture has changed to kind of achieve that goal that we specified before, where we can jointly model atoms and sequences. And the next thing is you need like a loss function that can kind of operate on all of these systems, right?
Right,
and so
before you go on, can I ask one more quick question?
Yeah, of course.
I really appreciate you taking these. So a very common covalent modification is like post-translational modifications.
Yeah.
So did you specifically identify and handle like phosphorylation, for instance, or some of these others that are acetylation, which are probably well represented in the PDB, as a sort of special test case, or or how specifically were these handled?
Yeah. So at training time, it's a little bit complicated, but basically the way the PDB handles this is that there's two ways you can kind of represent what I call a covalent modification. The first is that you have a different residue in the polypeptide sequence. So a phosphorylation, like you might have like a phosphotyrosine or like a selenomethionine or whatever, right? The second is you have a small molecule and that has a covalent record to some residue, right? And so we do train on both. I think, like, at least to me, like phosphorylations are like the modeling challenge with phosphorylations is how they've changed the backbone structure and less how well you model the phosphorylation. So for the evaluations we did in the paper, we kind of focused on the class of molecules where it was a small molecule with a covalent linkage. But yeah, and I guess this is another kind of reason to directly process things from the CIF files and not to use a pre-specified training set, because we can pull these information as we want to.
So just to be clear, for the modified amino acid case, where it's not like a covalently bound ligand, are you treating these as atoms, or are you modifying them to canonical residues, or like how are they actually handled?
Yeah, it's sampled stochastically, basically. So like 75% of the time they're treated as atoms, and then 25% of the time they're canonicalized. Yeah, just so that, this, as I said, we haven't really spent time optimizing this. There might be a nicer way to do it, but yeah. So, okay, so getting back into kind of the loss function, in AlphaFold, there's this introduction of this loss function called frame-aligned point error. And it's really nice for a couple of reasons. One is that it gives a strong pairwise gradient back to the network. And the second is that it enforces chirality of molecules. And the way it kind of works is that you take every NC alpha C triangle and you kind of perform this alignment on it. And then you compute the error of the remaining atoms with respect to that alignment. And then you can kind of get some equation that looks like this, where you have a bunch of frames and a bunch of atoms, and you have errors across all of them. And, they do some nice tricks like clamping large errors and normalizing the final value. And so but in small molecules, you don't have this NC alpha C local coordinate frame. So what we kind of intuited is that you could randomly choose. So, I mean, if you have a molecule, it has multiple sets of things that it's bonded in three ways to, right? So you could kind of just like stochastically sample these and, use that as your loss function. And the reason we chose not to do that specifically was just that we felt that we didn't try it, but we felt that it might be less stable, where the same molecule prediction could have a different loss depending on your choice of frames. So what we decided to do is kind of create a deterministic, or as deterministic as possible, algorithm that chooses a local frame for each atom depending on the bond geometry. So you can imagine, like in this case, we choose this frame, and they're just generate a set of rules so that every atom gets a chosen canonical frame. And then you can imagine applying FAPE is very similar to how it's done in AlphaFold, right? Where you have a predicted coordinate, this is a canonical frame for it, and then the true, and then you align these, and then you compute the errors with respect to that frame. And this is kind of the details of that. And the only difference really is that you have to gather the coordinates for each frame because they're not just in the NC alpha C positions. Yeah, so that's kind of the general. I'm going to talk quickly about a couple of results, but that's a lot of the structure prediction, architecture, and loss function information. Yeah, are there any questions? Do people have any other topics of discussion at this point before I speed through a couple of results? Cool. Yeah, feel free to. Oh yeah.
So sorry, quick question. So when you change representation, what exactly do you fit in neural networks then when the representation is not provided? Do you feed like special tokens? Say you don't provide 3D coordinates for this region. What you do? What exactly input will be?
Do you mean like in this atomization case?
Yeah.
Yeah. So you provide just the element types and the bond types and the chiral centers for those, right? And all of the coordinates at the very start are just initialized to like the origin functionally, right? So you don't provide coordinates in any case. But in those cases, if I'm treating these three residues as, sorry, can't see my hand, these three residues as atoms, I don't provide multiple sequence alignment or homologous template information for those cases. Does that answer your question?
So what exactly do you fit into neural networks? You don't provide this information, so what neural network gets as input?
It gets, for example, if you had glycine, it would get nitrogen, carbon, oxygen, or something like that. And then the way those are bonded to each other.
I see. I'm not sure I get. All right, let's move on. Thank you.
Yeah, I can try to explain it better. I'm not sure if I have a better slide for it, unfortunately. Okay. So, I mean, yeah, the first thing we're kind of concerned about, when you kind of do neural network surgery on these things is that we lose accuracy on the task that we'd started on. And so we were pretty happy to see that that wasn't the case. We have on protein monomer predictions, this is Cas14 targets. We have very, very similar accuracy to AlphaFold 2 and RoseTTAFold 2. So that's nice to see that we haven't broken it. So the next thing, I'm going to skip over some of the other results, but the next thing that I want to talk about quickly is kind of analyzing how the model generalizes. And so in these cases, sequence homolog means 30% similarity. So the negative case here is less than 30%. And then similar ligand is 0.5 Tanimoto score. So less than 50% of the bits of the Morgan fingerprint overlap. And, you do end up doing better on things in the training set than things that are not in the training set, but you do get a reasonable amount of things. And so this is for a small molecule docking functionally, right? Like flexible backbone, small molecule docking. Sorry, I really skipped many slides that are supposed to be in the middle here. But yeah, this is promising. At least there is a fraction of things that you do get predicted accurately, regardless. And the nice thing is that you can look at some of the examples that are dissimilar to the training data set, and we are able to make some accurate predictions here. And the really interesting thing, though, is that if you look at the correlation, so if you take the native complexes and you measure the Rosetta energy of those, right, the accuracy of RoseTTAFold All-Atom really correlates with how tightly Rosetta thinks that these two things are going to bind. So, I mean, it might be getting there in different ways, but the accuracy does correlate with some notion of our previous understanding of physics. So, and I think that kind of does explain, how the network's able to make accurate predictions on cases that were not seen in the training set. And so, yeah, then you can kind of put all these things together now. You can kind of get the protein, the nucleic acid, the metal, the small molecule all in one prediction. It's just like a neat case study that, this is now possible. And so, yeah, and then quickly on covalent modified proteins, this is kind of a similar analysis of how you do, depending on how similar protein sequences are in training. And, similarly, when you're really far away from sequences of training, you have much more inaccurate structures, but you still have a good fraction of things that are predicted accurately. I'm not going to belabor the point here. Then these are a couple of examples of some glycans. It's pretty neat. Obviously, I mean, these are cherry-picked examples. There are many examples that aren't predicted well, and there is more discussion about that in the paper somewhere. And then I'm quickly going to talk about, now we have all of this, and how can we apply it to, so we've painstakingly made training data sets, we've made architecture changes, and now we have to figure out how to do, can we do backbone generation with this? And so around the time that I was working on this, there's a lot of advances in diffusion. Sorry, this slide got very grainy. But basically the idea of these diffusion models is, I guess you discussed RFDiffusion in this group last week, but just to quickly go over it again, you iteratively add noise to your input. And in the case of RFDiffusion, it's Euclidean noise on the C alpha coordinates, and this Brownian motion of the manifold of rotations for the frames, until you get this like completely corrupted structure. And then the network learns to denoise this. And so now this is work done mainly by Ahern, but here's a trajectory of using the same principles that you guys discussed last week in RFDiffusion, except using this network architecture that can explicitly model small molecules. So in this case, the small molecule was given as a input motif. So it's the uncorrupted portion of the structure, and the residues and C alpha coordinates of all the protein were corrupted. And and then, you're able to diffuse this protein around the small molecule. And so kind of looking at some benchmarking cases, the top here is Rosetta energy. So the first gray, yellow bar is the using RFDiffusion to do this. And, it creates really unrealistic and unphysical interactions. Whereas, when you use this network, you get much lower energies and much higher consistency with AlphaFold. The other nice thing is that these designs are generally pretty novel with respect to the training set. So we kind of took all these benchmark cases and then took the TM score against the PDB. And so here the pink are things against everything in the training set. And then the blue is the training set with the same ligand, right? And you can see that they're actually really different than things with the same ligand. So it isn't just memorizing some likely binding modes. The last thing that I'll say on this slide is that, there's a concern oftentimes when you show novel structures that they're also not self-consistent structures or not real structures. I guess those are also two different things. But here, you can see this distribution looks exactly the same for the self-consistent structures. And so, yeah, so people took this into the lab. They designed a bunch of cool stuff. These values here are the TM scores against the training set. So you can see they're actually very, very different than things in the training set. So the first one is against everything in the training set, and the second one is against things with the same molecule or a similar molecule in the training set. And yeah, this is really nice. You can create these proteins that, yeah, have absorption profiles different than those in nature. So you could potentially expand the wavelengths of photosynthesis. So yeah, this is just a neat application of using this type of a model. And yeah, that's kind of the general ideas that I want to talk about. Yeah, if anyone else has any questions, I'd be happy to take them now.
Any other questions? Ajar, do you want to ask it?
Yeah. Can you hear me?
Barely. Speak up.
But there is a question in the chat.
Oh, sorry.
Okay, Ajar is asking if you can expand on the use of potentials to guide the diffusion.
Yeah. 1 second. I've lost. Oh, sorry, I pressed some button on my computer and I lost where my Zoom tab is. Let me just reshare. Yeah, so basically, go back to this slide. So the idea of using potentials to guide diffusion are at sampling time. It's kind of similar to the chirality feature idea, right? Where you take so you take the gradient of some heuristic that you have with respect to the previous step. And then in addition to nudging, so at every step in the diffusion process, right, you nudge the noise towards your final answer, right? And in addition to nudging the noise where the model wanted to nudge it, you also nudge it in the direction of where your heuristic function would like it, right? And so that's kind of the general idea. And then oftentimes in that case, you get much better alignment with like a protein designer's like intuition of how these heuristics should be sampled.
So he's asking if using the atom RF, if there'll be a better chance to have the sidechain rotamers correct. And I guess the idea is that if you're explicitly modeling the atoms, then maybe you're getting better backbones sort of qualitatively through that. Maybe I'm misunderstanding the question, but.
Yeah, I think that's a really good question. In this case, so like in this case, for example, where we condition on a sidechain, we're actually using the frame representation for the sidechains. We're not using the atom representation. But I think you're right that that is an interesting question in general. Yeah, I don't have a great answer for you.
So
Thank you so much.
I was curious if you could comment on the sort of the computational requirements of this relative to like RF diffusion. Does it take longer to train? Is the inference larger? Does it take more memory?
Yeah, that's a great question. The RF diffusion all-atom takes roughly the same amount to train from structure prediction weights. The inference is probably slightly slower because I think this model has more parameters, but not notice. It's not like order of magnitude slower or anything like that. And I think the memory consumption is probably pretty similar. It scales with length of your input. So yeah, I think people in the lab at least it's been roughly similar.
Any other questions? Oh, I see one from Stephen. Do you want to just ask that, Stephen?
Sure. Yeah. I was wondering, like, did you do any tests to see whether the SE3 property was like a necessary feature? Like, it's a inductive bias that kind of adds a lot of like extra architecture on top of the transformer. So did it improve performance like significantly?
For structure prediction?
Yeah, but also the diffusion process.
Yeah, I personally have not explored that specifically. I think like in terms of the diffusion process, we start with the structure prediction model weights. So you have to use the architecture used in structure prediction. So yeah, we haven't really explored that.
Okay, yeah, I was just wondering. Thank you.
All right, well, let's thank Rohith for a really wonderful presentation, and congrats on the whole team on the project. It's really impressive. So thank you for coming. And
Thank you. Yeah, thank you for having me.
We'll save the slides and the presentation and put it up on the web. And we look forward to seeing you in 2 weeks. Hopefully you all can join, and we'll be back for more. So thanks again for coming, and take care.